like an obstacle course.
Yeah, choose your own adventure path.
So the reason we're taking a water sample now is so we can compare the microbial diversity in the water to that in the sediment. And once we sit the ROV down on the sediment, the bottom gets disturbed and the water sample includes a lot of sediment in it. So we're trying to get this water sample first. And that'll allow us to compare the two communities and better understand what communities may be specific to the sediments here.
It's one of those instances where the jaw's not closing. Stand by, sorry guys. Just the jaws are having a hard time closing.
So those of you who might have missed the start of the dive, there was a bit of a technical difficulty with the manipulator arm. So the engineers and pilots have been able to sort some of that out, but it seems to depend on what angle the arm is positioned in can affect whether it opens and closes. Maybe something in the trigger mechanism that the pilots use that needs to be replaced. So
got number one, but number one's been fired.
Number one's been fired, yeah.
All right, I'll just pull them both.
Beautiful.
Two triggered.
Two popped, yep.
Beautiful, thank
Roger
you.
that.
nice. Well done.
Now it's wanting to close. So interesting.
Quantum jaw close.
Oh, curious. All right.
So Lisa, I don't know if you can hear, would you like more than one core here?
Paul, I actually don't need any because, you know, we need to work in batches of 3 or whatever. Take it for yourself.
Okay, thank you.
Anywhere around here look good for you there, Paul?
Looks great. I don't know, Greg, if you see any critters that you want to try to capture in the process.
Yeah, I don't see the anemones here. It's anywhere here is fine.
Raj.
Hopefully it's deep.
So if I recall correctly, it will be these two here on the right
Yes.
towards the camera.
Yes. The middle one on the starboard side is open, and the back one on the starboard side.
Roger that.
Mind if I do a Argus zoom?
Yeah, go ahead. All right, now we can go. Also to calibrate the wrist rotate too.
Super band.
Super band.
Yeah, so if you haven't seen one of these before, the rubber bands to help keep the push core from floating out of its quiver while
Goodness
the sub's
gracious.
diving, and it's supposed to break.
They're very, very strong.
Should be sponsored by that company.
There you go.
Wow.
All right.
If we could get some captures of them taking a
Yeah.
core, that would be fantastic. Thank you.
Great idea. Looks like she'll go. Oh, maybe I spoke too soon.
Go.
Actually, I spoke way too soon. Sorry. We're actually moving the
Oh, there you
vehicle
go.
now. Oh no. That was not a good quiver. Not a good core, rather. All right, well, it's not terribly deep here. Want me to redo this one, or?
Yeah, if it won't go down further, we really need only 5 centimeters. So it's not entirely clear to me how deep that one penetrated. If you want to pull it out and it looks like we've got 5 centimeters or more, we'll keep it.
Roger that. And try giving it a little bit of a tap. Yeah, it's as far as it'll go.
Okay.
Jonathan, can you push in there, please, on the HT bit?
I can, yep.
Pull away, Jonathan.
Go ahead. Oh,
Yeah, it's not really that consolidated there, hey?
It's going to come out.
Yeah, do you want me to just throw it out?
Yeah, please.
Raj? All the way up in my bar here. We can try one more spot, but it seems like it's relatively shallow here.
Yeah, if you want to give it one more try.
Roger that.
Oh.
Maybe.
nice.
Going. Oh, that looks good.
Ooh.
Might have found a deep
Nice.
spot. Nice.
Oh yeah, get
done.
in the sediment.
Well done.
Beautiful.
Oh. All right, we got one. Push on in there a bit, please, Jonathan. That's great. A little wide, please. That's good.
Hi science team, this is Kira from the lounge. I saw that you guys did a Niskin and now a nice core from here. It could be cool to also have a correlated rock if there's anything nearby of interest. Just a thought.
Oh wait, Jonathan.
We'll have a look. It looks all pretty solid without any free rocks, but we'll have a look around.
Oh, no. So loose.
Sediment's
Yeah.
not consolidated,
farther.
unfortunately.
I don't think that one's sealing. Like,
Yeah,
seems like as soon as you pull it out, it's just.
Just falling, yeah.
Yeah.
Might be
Yeah.
still some in there. It's kind of hard to tell.
I don't think so. And plus I jostled it when I hit the light. Huh. Sorry, guys.
It's okay.
That's
It's
okay.
okay. We tried.
It did look like it wasn't sealed.
Yeah.
It
Yeah, there's
started
Yeah.
falling out
Want
immediately.
to try another one?
I think it's not clay enough. It's maybe too coarse. The suction might be a better bet to get a sediment here, but yeah, I think we can move on.
Raj.
Thank you for trying.
Oh, of course. I don't really have a good angle on bubble there, do we? I think we're good here now.
Kira, again, no need to look for rocks here if you guys prefer to move on. Thanks.
Okay, thanks, Kira. We will.
Make sure that light's still good. That's right. All right. Well, sorry, guys.
Do we have the digital stills cam still mounted?
Yeah.
Still taking pictures.
Nice.
I got myself hung up on a light there. A little bit. I'm going to push in past those little rings there, Jonathan. All right, and go ahead and for the bumper there.
I'm going to call ahead.
Roger that. Jonathan, are you still zoomed in on Argus?
A little bit.
Raj, thank you.
So Kira, are you watching from the lounge or are you just listening from the lab?
It looks like there are a bunch of rocks over here.
Or maybe neither?
Yeah, they're a bit angular. I think she was asking for some that were more lobey.
Raj.
So we'll keep going unless she chimes in. And we're not seeing a lot of biology on them.
Yeah.
Do we know the name of that fish?
It might be an eel pout. Not great with fish. It's like we've had a bit of a slump there. We've got a couple of, there's a bracingid sea star there perched on the rock. Yeah, that rock's nice and lobey, but unfortunately it's all one big
piece.
mass.
Yeah. Yeah. A little difficult to pick up. Steve noted that we passed over a thorny head fish.
Yeah, the
That
red one.
orange fish. Yeah. Very stark contrast.
So what is that coral on the right there?
Oh, yeah, good eye. And thorny heads are a type of rockfish.
Oh, there's a coral. Well spotted.
Yeah.
Wow. Blending in there.
There's an octocoral in the frame. Oh, I think it's
Oh,
a sea pen.
it's a
Sea pen
sea
with
pen,
the rock peduncle, the one that doesn't have to.
Cool. All right, Jonathan,
And
go ahead and push in there, please.
It's the one that forms the question mark, and you can see its peduncle, its base. So, Paul, you haven't had a sea pen.
No, we haven't.
Might be a rock pen.
Rock pen.
Rock
A
pen,
rock pen. Anthopatillum.
Yeah.
Interested in
So
a
sea
sample?
pens are normally...
Do you want the sample or?
Yes, please.
Okay.
Pull away, Jonathan.
Sea pens normally go into sediments, and this one appears to be attached to a rock.
Don't they usually retract?
No, but the polyps will pull in.
Just the polyps, okay.
Yeah. Yeah, they've still got a proteinaceous rod up the middle, like other gorgonians. So Steve Alskovich, our resident coral expert, thinks this is Anthoptilum. Thank you, Steve.
You would, Jonathan, push in full, please.
Yeah, it's definitely attached to a rock with
Yeah.
a very
Oh.
distinctive peduncle.
Ooh.
Video, it's wet lab. Yeah, I tried to text Al, but he didn't answer me. Maybe you could try. I'm in the wet lab if you could come and help me. Thank you.
That's very cool.
Before you go video, full wide, please.
So what do you think? Slope? Or too big to be sloped?
With kind of my understanding of these, the little hook part or the question mark part is a little bit
Rigid?
rigid. Yeah, it's like almost a spring.
Okay,
So it might
so
be
to jam
flat.
up.
That'd be kind of my, I know we have slurped some in the past, but they are relatively stiff.
It
in
is
that
pretty
case.
big. Yeah, so
Yeah,
we can forward by a box.
Roger that.
Yeah.
Given our luck of getting things stuck.
Yeah.
Mhm.
yeah. Yeah, that shape might just clog up.
Lesson learned.
Yeah. You want to push in there a bit there, please, Jonathan? That's great. Thank you.
Go ahead, Bridge. Thank you.
Mhm.
Wonder if you grab some of the rock at the same time. It would come up.
Looks like it's part of a pretty big rock, unfortunately.
Just
See how good its peduncle works. Oh, wow.
Nice
Yeah,
section
if
there.
I remember right, those are pretty cemented on.
Yeah, you can see the distinctive protein rod that holds it erect, and then it's going to this sticky kind of expanded base, which is not a skeleton. So maybe it's just stuck on there.
could you scrape it off? You just scrape it off at the base. Yeah. Yeah, exactly.
Oh, nice. Once they're stuck somewhere, can they move?
I don't know. Mostly pens stuck in the sediment won't. Some can really close up. But I don't know if they can migrate around.
Hopefully it won't float away if we scrape it.
Yeah, it's hopefully not. I can just get a little bit under him.
So it would be good if we can get this peduncle. Yeah, it's lifting. Good job.
It's coming.
It's hardly the ideal tool to pry that off.
Yeah.
Yeah.
Oh,
Nice.
Yeah.
well done.
Super rapid. Nice.
Nicely done, Jess.
Thanks, guys. Before it floats away.
Yeah.
Yeah.
Oh, wait,
That's
Jonathan.
it.
Got low enough in the current, it didn't take it.
So we're going to put this in the forward bio box on the port side, number 6 white, and this is going to be sample NA124 074.
074.
All right, go ahead, Jake.
Oh no.
Raj, go ahead and pop it out slow. Yeah.
Hopefully it sinks on in there. Nothing floats out.
Fantastic.
And it goes.
Little halo.
And it sinks. That's good.
I think it'll probably start jostling around when we close it, so it'll probably sink.
Awesome.
Cool.
Nicely done.
Fantastic.
Thanks, Jake. All right, ready.
Roger.
Jonathan, you want to push in past Mongo, please? Thank you. Rennie, how far along on our trek upslope are we?
We have 770 meters remaining until we get to the top point V.
Raj.
And I think if we go at point 3, it'll be about an hour and 15 minutes with 0 stopping. And it's 145 right now, 140. So conceivably with some stopping, we'll be able to reach the top.
Raj.
But not too much.
You know, Greg, if we're able to come back on the Falcor, we should plan a dive where we put out one of your minnow traps and then do another dive the next day and collect it, see what we catch. And
Yeah, we can close it up.
we could get some of these shrimps.
Some of these
and who
shrimps,
knows what else.
very
Yeah,
likely.
Yeah.
Saw a bunch of these shrimps yesterday. Still seeing a bunch of them today.
I was mentioning that Nautilus actually did a dive on Northeast Bank on the west side, and we climbed. It was a 24-hour dive from the base, climbed the slope, and then we were able to spend a bit of time on the top. So it'd be interesting to kind of see what that dive yielded, to look through those photos and compare it, I guess, to this slope, and whether or not we get to the top, see what.
Yeah, so we have 700 meters to get to the top.
700 meters horizontal, yes.
Yep. $750.
Well, we've only covered 200 meters in an hour and 45 minutes.
Well, that's vertical. But
We
Yeah.
started at that 070 was that sample we took, so we have covered around 400 meters.
Okay.
Horizontal.
yep, we'd have to speed up. Is it even possible to get to the top?
It is.
Okay.
If we go 0.3 knots, maybe about an hour and 20 minutes when we're going 0.3.
We'd have to really motor and not stop. Well, if we get a few rocks here, maybe we could speed up a bit just to get through the OMZ and into higher oxygen. But we should get some rocks for this around this region if we can.
Sure. Let us know when you see good ones.
Thanks, Jake.
I wonder if the faster it is. So now we're doing this.
Lots of sediments here, like covering lots of those rocks that we've been seeing. Still seeing some of that bubbly rock texture. Some of these large outcroppings of rocks. We're on the hunt for some smaller pieces of rocks that we would be able to pick up. And we're looking for certain types of textures that could be indicative of crusts that we're interested in looking at, both the macrofauna, the microfauna, the microbial communities of those crusts, and even the mineral composition. Got a whole bunch of scientists collaborating together to give us a better idea about some unique crusts and the minerals that are on these rocks and what could be living and the ecosystem that they're part of. We're currently at a pretty low oxygen level right now, at around 5 micromoles per liter, which is pretty low. As you can imagine, any of the creatures that we're seeing here are living in low concentrations of oxygen. We're at a depth of around 130 meters.
These rocks look pretty dark with a possible sheen, characteristic of a ferromanganese crust.
It's definitely one of those types of crusts that we're interested in.
There's a bunch of kind of looser rocks over here. Would you guys want to look at these?
Yeah, let's see if we maybe can
Jonathan, you want to
pick
do a
up one or
partial
two.
zoom?
A bunch of these Faria sponges here too.
These look like viable
Yeah,
candidates.
I think we could.
These look like good candidates.
Okay.
Many different
Great
rocks.
snap.
Yeah, these look great. Cool, by
Exactly
Jonathan.
what we're looking for, not those really large ones.
Hold position. Thanks, Vitali.
Hi guys, Kira here from the lounge. I agree there's some good candidates here. If we can go for ones that look more bumpy and a little less smooth, that'd be great.
Yeah, bumpy rocks heard.
Bumpy but not angular. Is that correct?
You got it.
Round and bumpy. Interesting parking position in here.
Steve confirms that these are, yes, these are Faria sponges.
Let's try again.
Live and dead ones, correct, Steve? The yellowish ones are look dead.
Covered with some nice sediment. Live or white? Yes, says Steve. if we're taking a look at these rocks.
I think we're okay. It just looks scary.
Rog. All right, go ahead and push in a bit there, Jonathan. That's great.
Okay.
One with bumpier texture.
Here, are you seeing any that are sticking out to you?
Yeah, in the view you are right now, the one that's about 11 o'clock, it looks almost like slanted, not the big blocky one.
Right. I think I know which one you're talking
If
about.
you use the pointer screen in front of the main scientist position, maybe I can...
You gotta show me
Yeah,
Not sure you can see that down in
I'm
the...
pointing it, but
Oh, you're right, I can't see it. Yeah, the one
So-
that's down from where the lasers are now.
yeah, let's down from the laser and a little to the left. Correct?
Yes, exactly.
Okay.
It's got that nice big bubbly texture, botryoidal.
Okay. I think we're talking about this one.
Roger that.
See if that's the right one.
Let us know, Kira, as we get closer.
There's a crab in the back checking out what we're up to.
This one here?
Is that it, Kira?
Yeah, it looks great, thank you.
Awesome.
If it's not too big.
It'll be all right. I don't want the second one.
Two for one.
Oh, no, it won't close. Stand by, sorry. Okay.
Everybody's watching what's going on.
Yeah, we've got like a second audience here.
I know, they're probably not very happy.
And photos of that context would be great.
Absolutely.
Sure thing.
Definitely getting lots of screen grabs.
Push in there a bit more, Jonathan. That's great.
We're gonna be famous, Rock.
Wow.
Ooh.
Bottom
Very
almost looks
cool.
red.
Yeah.
And then it's got that bumpy texture on that top. Really nice crust.
Yeah, it looks like iron alteration where it's in contact with the sediment. And more manganese enriched on the top. But yeah, it looks like some good biology associated too. Hopefully some microbes.
Full way, Jonathan. All right, so where are we wanting to put this guy?
Let's go ahead and aim for the starboard bio box.
Rog.
We'll aim for number 4 green, which is the starboard forward box
Roger that.
on the outer side.
Prior to doing that, I'm going to back up since
Yeah.
I'm kind of
Parked in
pinned.
there.
But we'll just kind of hang out with the rock here.
Kira, is Lisa there with you?
She's down in the lab. She's got audio but not video.
Lisa, wondering if you'd like a second rock from here or whether we should go somewhere else. Well, we flipped it off, so we'll go somewhere else.
Let's see. Can you hear me?
Yes.
Can you hear me?
Yes.
guys?
Yes, we can.
Yeah.
Oh, now I can't hear her. Nope.
Video wet lab, you might be cutting in and out. Don't hear you.
There you go. Now you're good.
Can you guys hear me?
Yes, yeah.
We can hear you and the construction.
I would be happy to get rocks further up the slope. And
Okay.
Bob did offer us to stay in a little longer today.
Okay, we'll put this rock away and then we'll move on.
Did you guys hear that?
We
Yes,
hear that.
Yes.
We heard that. Thank you.
Acknowledge.
Yeah.
No.
How is it on the right side?
Cheer.
All right, and is there anything floating in the starboard bow that we should be aware of?
Nope. That box should be empty. All 4 boxes back there is empty. That entire area should be
Gotcha.
empty.
Sounds great, thank you.
Okay, now you have SPL.
Okay.
Switching over to the sample salvo.
Can you guys hear me now?
Yes.
Yes, we heard you before as well.
Oh, you did. Okay, great. Thanks.
Go ahead, Jake. Any preference, Science, or just anything we got?
Yeah, whatever's closest to you. Let's see, which one is that?
Aft inboard.
Aft inboard. Beautiful. Thank you.
It's a little loose in there, but yeah.
Coming in.
Aft inboard is starboard bio box number 1 blue. This is going to be sample NA124-075, and I believe this is rock number 2, so R2.
Copy.
Beautiful. Thank you.
Yeah, no worries.
So we're going to keep moving up.
Yeah, we'd like to start another transect.
Sure thing.
Roger that.
And you could, if we want to move a little faster, we'd be happy with that too.
Rog.
We won't stop. We'll try and get some distance covered.
Roger that.
Copy that. I'm going to start moving 305, and then we'll start the transect as soon as Argus starts moving in your direction.
Rog.
This is going to be transect number 3.
I'll get closer and things like this. I'll just get a little in front of Argus.
Yeah, copy.
We could start it. All right. Well, maybe I'll get over that little lump.
So welcome anybody who's joined us recently. Happy Halloween. We're off the coast of Southern California.
I'll start pushing forward.
On what's called the northeast bank, and we're on the eastern side of it, ascending up to, hopefully we'll get up to about 500 meters. We're currently at 825 meters. This northeast bank was once an island. It is an ancient volcano, up to 11 million years old. And the surface is flat on top if we get up there, and it's where it was once an island about 90 square kilometers. We're interested in the rocks along the side. of the flanks here that have accumulated crusts of ferromanganese or phosphorites, and how those crusts are formed, and also in the biology, the animals and microbes that are associated with them. So we'll be sampling along the way.
That's fine.
We started off at the bottom of the slope
Gotcha.
with about 1,200 meters where there was lots of sediment. And we're seeing now a mixture of mostly rocks with still a lot of sediment as we come up. As we're ascending, the oxygen's been dropping. And when we get to a certain point, in a few hundred meters, it'll then start to rise again.
Sorry, we might have missed it in the back row. Did we get a confirmation of when that transect started?
We did not start, and we will start now
Oh,
because Argus
perfect.
just started to move.
Okay, thank you. Just
I
thought we
answered
missed
two questions at once.
it.
Yeah.
And that was our fault not recording. Thank you.
On each dive, we try and do a few of these transects where we stay a pretty stable distance above the bottom with the two lasers there. They're green lasers that are 10 centimeters apart, and we run them for about 100 meters, and that allows some scientists to then quantify the distribution and abundance of organisms along the slope. So we've done, this will be the third one we've done on this dive. We'll probably try and do another two before we finish.
There's another Panichia, deep sea cucumber, deep sea cucumber.
Ooh, a bunch of different critters on that rock face. One of those Pharea sponges, that white, glassy looking sponge.
Lots of little squat lobsters. They're not really lobsters that people might be familiar with on the east coast of the US. They're actually more closely related to hermit crabs. We see a lot of squat lobsters in the deep sea. People in California might be familiar with a kind of squat lobster that washes ashore every now and then in abundance, are the tuna crabs. But most squat lobsters live in the deep. Often they're reddish like this, or in the really deep sea we find them that they're white, living around methane seeps or hydrothermal vents.
Do all these different species, Greg, prefer low oxygen?
They can tolerate low oxygen, and the tuna crab, in fact, we found in abundance in very, very low oxygen in Costa Rica. Very large sea cucumber there that we haven't
Looks a
seen
little
before.
different.
Yeah, it's not a Panichia, it's another kind. There's another one.
There's another one, yeah.
Could be a Synallactes. But we're on our transect, so we're not going to stop.
Kind of in a different looking rock rubble field here.
Greg, are those white tuna crabs that you were talking about that you found in Costa Rica at methane seeps Yeti crabs?
We found normal tuna crabs, red, at an oxygen minimum zone in Costa Rica. It was, there appeared to be some methane, oh, there's a sea whip. There was some methane seepage there, but the seeps were much deeper, and there we found normal squat lobsters that are in the, there are two kinds, Munidae and Munidopsidae. And the Yeti crab, which have hairy arms, and they use those arms to farm bacteria, are another group. They're related to squat lobsters, the other squat lobsters that are orange or white, but they're in their own group called yymmdd. And the first of the Yeti crabs was found in 2005 at a hydrothermal vent in the eastern Pacific. And that one was pretty big. And at first, the original scientists who named it, Kiwa hirsuta, which means hairy arms, thought it was a carnivore because it was such a big and menacing looking crab. And then the second one that was found at the seeps off Costa Rica was found in big assemblages around where the methane and hydrogen sulfide was flowing out of the sediment. And the crabs were actually waving their arms backwards and forwards. You can find some videos on YouTube of these, if you look up dancing Yeti crab. And this movement of the arms was then a clue to some scientists who then investigated the hairy arms of the crab, and it turns out they're farming
So a couple
bacteria.
observations here, sorry, Greg. So we've seen our first sea whips, we've seen our first bubblegum coral.
The Paragorgia bubblegum coral.
The first sea whip was around 800 meters, and this is about the same depth that we started to see them yesterday, at least in higher abundance yesterday.
We saw another mushroom coral.
So I don't know, Steve, if you think some of these octocorals, these gorgonians and whips have a lower depth limit, because we definitely noticing that we don't start to see them until we get up into the shallower range. But hopefully by the time we in this transect, they're in a little higher abundance and we can get a couple.
Yeah, we're going a lot faster on this transect, so it's going to be pretty hard for us to stop it all.
Those are these sea stars, Brichanthia. Just passed over.
Which one, sorry, Caitlin?
They were like the red, like the one we're seeing on the left
Oh,
now.
that's a Brisingid.
Brisingid.
Yeah,
I was getting them mixed up with the Pluricanthia. Brisingid.
Yeah, that's a sea star that eats from the water column, so it holds those arms out in the water column and has sticky, very sticky. If we collected one, they usually break up when we grab them, and the arms are incredibly like almost like Velcro when you grab them. So any little plankton that comes by is not going to get away from those.
So a bunch of little mushroom corals there.
Oh, yeah.
Oh, yeah.
Really cool colors on them.
Big synallactid sea cucumber there.
Another beautiful Brisingid sea star. Really cool to see those sticking onto the rocks. Is that another sea whip there?
Yeah.
15 meters remaining.
Beautiful,
Roger.
thank you.
Should try for another rock after the transect. Take care of some of Kira's sampling. We're thinking about you, Kira.
We our first Swifty are on the right there. Red
Oh, yeah.
branching soft coral.
All right, Lisa tells us they have video now in the lab.
Nice.
Good news.
Need to come up faster here. Coming up faster.
Yep.
Oh, there's another sea whip.
Come up there,
Another
Jake.
sea whip.
Yeah, it's pretty steep there.
Yeah.
Come up. That's the end of transect, actually. So we'll
Beautiful, thank you.
fly up there. Do you want me to stop up and we look for a rock?
Yeah, let's stop
Yeah.
the ship here.
Bridge nav?
Yeah, that's a good idea. We'll try and pick up another rock.
Hold position. Thank you.
Although it's,
We'll just get up this steep wall.
yeah, no rush at all. It's,
Taking a look at our Argus proximity.
maybe we could just sort of continue on. If we see a sea whip or another kind of coral, we'll settle down there and pick that up and a rock.
Sure.
Roger that.
Looks like there's a few of something stalked. This could be those Aspestopluma Sponges, though. I think that's what those are named, the carnivorous lip-like sponge.
Oh, we got one here.
There's a whip on the right.
Yeah.
Oh, yeah.
Do you think we're still swinging though? Looks like we're
Probably pretty settled out.
Yeah.
I'll keep an eye on it.
And some rocks in
Probably
front.
got some vertical up. You
Yeah.
need it 17 delta.
Raj.
I can back up too.
I keep an eye on the sonar.
Yeah, Raj.
Looks like there's 2 sea whips here. I can get a small piece of one and then slurp it, or they might be a little floaty.
Yeah.
Could be hard to collect.
Yeah.
It's not going to be easy to break that off.
There's a bit of a smaller one back there.
Can you reach into that one?
Yeah.
I think, or
Yeah, we can try that one out.
is it risky to try and slurp that one?
It seems relatively smaller. Go ahead and push
Yeah.
on in there a bit there, please, Jonathan.
It's hard to tell how long it is.
Yeah, it's kind of out at us, isn't it?
Might have a bubble view of it.
Yeah.
Another angle. There you go.
There we go. 10 centimeters
See it
about?
over there.
Yeah.
10 centimeters? Oh, it's
Yeah.
not too big. It should come through the slope.
Yeah. I think it's worth a shot. All right,
Is that
Jonathan.
big enough, Paul? Or you, no, you want the bigger one.
Big one's better.
We could, maybe we'll get them both.
Raj, then maybe I'll just trim the big one.
Paul would like the bigger one because it's better for the chemistry.
Oh, Raj. Okay, so the larger one here.
Yeah.
Roger that. Wonder how easy it's going to be to break. Yeah, we can try anything out.
You get all of it in 2 pieces in the slurp, maybe.
Yeah.
Feed
The
it
big
to
one,
the slurp. Pretty hard without those coral cutters, though.
Yeah. Some of those coral clutters would be really nice.
Yeah, this is the coral smasher.
So Steve tells us this is a primnoid octocoral, possibly Norella.
Steve says it'll be pretty floaty.
Yeah. Go for the midway point here.
You're out of auto XY, so I'm just going to give it a DBL reset while we.
Roger that.
Very
Okay,
good.
We got one piece.
Awesome.
Jonathan, go ahead and push on in there a bit, please. Great. All right, so we'll take this guy
Thank
here.
you.
No worries. Little wide there, Jonathan. A little wider. All right, that's good there. All right, Jake,
Flush.
go ahead. You might get some sediment in it. You can just do 70% again if you'd like.
7%.
Reg. And what sample jar will this be going in, Science?
Can we aim for jar number 6, the coarse one?
Sure.
Jar number 6.
All right.
All right. See if I can get my jaws under there.
All right. Gonna get